I will just like a second to find the real one. I'm sorry, I had like 100 students today.
No problem.
And yeah, okay. Now mostly I work with WebEx, so let me see whether I can do it with your sharing screen. Da, 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 da.
It's the arrow in the little box next to the microphone icon.
Okay,
The upper right corner.
just press that.
Yes,
No,
press
this is,
and choose the desktop or the presentation directly.
or how about another one started now instead of me?
Don't
Uh,
worry, if you prefer, I can share your slides.
Please share my slides, it's
Okay.
easier. You just have to, but you cannot not use a mouse. Doesn't matter. You can share my slides, please.
Okay.
So,
Kusum, is everything okay? Can you see my slides?
Yeah, perfect. Go ahead.
Okay. So Branislav, you can start, and I will
yeah,
try to show with the mouse everything you point out.
yeah. You picked up here a couple of nice. Like, thanks for doing this job for me. I decided actually we did a lot of work. I decided to stick on lipid metabolism, which is our main focus here, and to compare basically here how the cells look like if you load them with triglyceride versus if they are loaded with lipid from some other reason. Maybe it's good for you to see how it looks like when lysosomes are full of lipid instead of lipid droplets, which we classically have. The first slide you see now, and if you watch the movie, you see the cells after 24 hours loading with triglyceride or VLDL loading. This happens basically in atherosclerosis. In macrophages, the cells you see here are primary peritoneal macrophages, so not cell culture, but primary cells. 24 hours is like you see quite a lot of lipid is accumulating in cells in form of lipid droplet. These are these clearly visible white dots. After a while, yeah, exactly. After a while, if you go to the next slide, like I think it's 12 hours or then 16 hours, you see more and more. And if we focus on the cell in the middle, it's good to see that actually after some time they died because it is already so much of lipid inside the cells cannot handle it anymore. Interestingly, they do not activate any other lipid digestion method like lipophagy. For me now, for you, not that interesting. What is interesting for you to see that the triglyceride or lipid droplet looks pretty clear, like a pearl, growing pearl, which remains nicely and quite clean, although there is so much of the signal inside. I presume you all have this experience, and if you have a lot of signal, that your cells are not looking that nice anymore. If you now switch to the other slide, please, you can compare this now with mostly cholesterol ester filled lipid. Is it now the case you can show this cell in the middle? There is one cell full of lipid in the middle and maybe one more on the right side. Doesn't matter. You see here actually a combination of a mixture of primary cells. Some of those are knockouts. They have inactive lipase, lysosomal lipase, which as consequence have accumulation of lipid in the lysosome. So for me, after watching many of those slides, it looks quite different. Now I'm not so sure, isn't it? Because lipid accumulates in the lysosome, where you of course have one other full membrane, which is, I don't know, carboxylated and everything else. Probably this makes it more blurry than the classical lipid droplet. Or maybe this is the case because actually here we see much more cholesterol ester than triglycerides. I love this video. We tried actually here to do some enzyme replacement therapy to mix cells with healthy cells. So theoretically, they should take over enzyme which is missing and picked in the cell. So what I expected actually that this foamy cell lose the lipid content. This is not really clear. I have to continue this work. But I picked up this slide actually because what you probably already saw, there's a small cell running around now, left and right. I called it by my presentation here a pet, exactly. The question was like whether macrophages have pets. If these are primary cells, this is very friendly thing. Normally macrophages eat everything what is not welcome there. Obviously this guy is very welcome. And then I realized after a while, oh, probably it's T cell, whether T helper cell or tumor killing cell, doesn't matter. But according to the size and the how it looks inside, it is, I would say, clearly a T cell. So another example from the same hypothesis, actually, yeah, exactly. You have it in the zoom in the last image I'm showing here. I'm quite sure it's T cell, and this is really huge advantage for the whole cancer research. I already suggested people here in my which work a lot with cancer to do some mixing. We can easily trace them. We can clearly see what they do. They obviously check MHC receptor on macrophages, so this is quite promising for immunity and immunological research. We tried actually here to get rid of lipid if we mix with healthy cells. Basically, I saw it after some days, but still I could not find a nice cell where I clearly see one. That T cell in the middle is, losing really lipid. Actually, after some time, I think T cell has died as well. So, it's probably already more than 5 minutes. I'm sorry, please interrupt me. Actually, I thought what we have to present here, or we should share actually basically what we do in the lab. So if you have any question, please help me to answer it. Thank you.
Thank you, Branislav. That was quite interesting. Does anyone have any questions for specific to these applications? You can feel free to unmute yourself.
So I don't know if maybe I could ask you all, well, if you ever follow lipids, whether you also have this experience in lipid-loaded cells or anything like that as extremely blurry. So it's really hard to get the clear image. The clearest I got was the previous slide with VLDL loading with triglyceride. I'm afraid I cannot get it any better.
I think Sanela, it might be about the slice exportation.
Is my microphone on? Yeah, we had several customers that were working with lipid droplets, and they have different drugs that are affecting the size of the lipid droplets, and and also they were using some acids, I think, to accumulate the number of lipid droplets in the cells. And we have some customers, I can share the publication with you, where they developed a tool to track lipid droplets, and I don't know what. Can you hear me?
I can hear you.
Yeah, so they developed the tool to track the lipid droplets and to calculate their dry mass based on the refractive index. So Nanolife is also working on developing this tool. So hopefully soon it will be available for the people that are working in the field of lipid droplets.
Okay, thank you.
If I can interject quickly, I was actually working with lipid droplets using a little bit older generation of Nanolife microscopes, like 2 years ago, and it's pretty straightforward to measure a couple of parameters of lipid droplets, including their velocity and size, and sort of based on settings, and also depending on what is exactly the types of cells that are used, you can get pretty nice pictures. And I was doing up to 24 hours of imaging, so doable.
Okay, thank you.
Okay, thanks for that input. Maybe we can then move on to our next presenter, which is Ira.
Yeah, thank you very much. Sanela, could you please share my presentation?
Sure.
Yeah, thank you very much for inviting me to participate in this meeting and to present some our data, which could be interesting to recognize this is the wrong one.
Sorry?
Yeah, thank you.
It's okay.
Probably our applications and probably our activities could be interesting for the seminar participants. And first of all, I would like to give you some information about our research interests. The Laboratory for Experimental and Translational Research on Radiation Oncology is interested in the elucidation of morphological and molecular features of carcinoma cells with increased metastatic capacities and radiation resistance. We also work on chemoresistance, but since we belong to the Department of Radiation Oncology, radiation resistance is the first kind of interest. Additionally, we work on the metabolic activities of cancer stem cells, revealing metastatic properties and therapy resistance, because we generally believe that cancer stem cells could be a reason of radiation resistance and chemoresistance. And additionally, since we would like to overcome both kinds of radiation and chemoresistance, we use different agents to kill therapy-resistant carcinoma cells, and therefore we would like to observe how our radiation-resistant cells demonstrate any response to ionizing radiation and chemotherapeutics. Next slide, please. As an example, we have compared head and neck carcinoma cells with the different radiation responses. Phagoparental cells demonstrated better radiation sensitivity, and radio-resistant phado cells demonstrated radiation resistance. And as you can see here, there are some differences in morphology between the parental cells with good radiation response and radio-resistant cells demonstrated a formation of vacuoles, different forms and different mitochondria distributions, and therefore we would like to know more about the metabolic activities of radio-resistant cells, since we see that mitochondria are fully different in radio-resistant cells and parental cells. Additionally, we are also focused on lipid droplets, and you can see also here the differences in lipid droplets between parental and radio-resistant cells. And we also observed the dependence of therapy response in the cells with a different number of lipid droplets. Next. Since we possess the 3D Explorer Fluor, we can also use fluorescent dyes. Either we could use conjugated antibodies, or we could use just the dyes to mark different organelles within the cells. And again, we have compared parental phado cells and radio-resistant cells stained with JC1. This is dye which could demonstrate the mitochondrial potential and also ROS production in the cells. And ROS production is very important for radiation response and for chemo response. Therefore, this microscope could be used as an additional tool to confirm our previous data received using other machines and other methods. And again, you can see the differences in mitochondria distribution in parental cells and in radio-resistant cells. And although we can observe rather fusion of mitochondria in parental cells, radioresistant cells, demonstrated the fission of mitochondria. And furthermore, you can also see the differences in mitochondrial potential. JC1 is not very good dye for mitochondrial potential. However, we could also observe not only the mitochondrial potential, but also the inclination for ROS production. And this is a quite useful method to use it using the 3D Explorer Fluor. Next. Since I said that we are very interested in cancer stem cells, we also work with differences in morphology in radioresistant cell lines. And radioresistant cell lines demonstrate the giant cells, and currently few teams, mostly in US, work on the giant cells, and it is generally believed that giant cells could produce cancer stem cells, and therefore the physiology, morphology, biology of giant cells are very important for us. And as you can see here, the giant cell is so big that the field doesn't contain the full cells. However, this giant cell provides us with the possibility to observe different organelles, and we plan to treat the cells to select the giant cells and then to see how the giant cells could demonstrate response to different kinds of therapeutic approaches. And as I said, that morphology could be very clearly observed in the giant cells, and you can see the tubes of mitochondria, a nucleus, nucleoli, nuclear envelope, lipid droplets, and also we see the vacuoles. And what is also very important in our research, lysosomes, because lysosomes also produce ROS, and if we will stain the lysosomes using different dyes, then we could also see the dynamics of the lysosomes, and we can see the behavior of the giant cell, including such important organelles like mitochondria and lysosomes. Next, please. Sorry, could you please?
I moved it to the next one, but I don't know if it arrived yet.
Okay, sorry.
Kulsum, can you see the videos?
Yes, I can see them.
We see
We can see them, yeah.
And I don't see.
Maybe try and go back and come forward again.
Go back and let's see. Now?
Yeah, now I see.
Okay,
Thank you very much. And next, our interest, since we work on different responses of different cells with different sensitivities to radiation treatment and to chemotherapeutics, we also use novel agents which could kill therapy-resistant cells, and we would like to see the differences in morphology and in the behavior of cells when they reveal different sensitivity to different compounds like elastin. This is a ferroptosis inducer, and little is known about the changes in the cells upon treatment with ferroptosis inducers. And here we have observed that DMSO didn't affect the biology and physiology of the investigated cells, and SCC25 head and neck carcinoma cells are quite sensitive to Erastin treatment, to ferroptosis inducer, and you can see that Erastin could induce cell death development. What is also interesting that upon the cell shrinkage, we could also observe the production and the release of vacuoles into the surrounding microenvironment, and these vacuoles contain also the cellular material. What is also important, probably it participates in the cell-cell communication, and they provide additional signal for other cells that they should die or they should survive. We have to work further on it, but finally you can see the development of cell death in the Erastin treated cells. Next. This is not very sensitive cell line to Erastin treatment, and if you will see, the cells demonstrate just shrinkage. They are becoming smaller, but only one cell demonstrates apoptosis development, and all others, it seems that they survive upon the treatment, and other methods confirmed our observations here. We observed that after the treatment, CAL27 cells could be recovered, and they recover very successfully, and therefore this observation is quite interesting for us, and we are very happy that we possess such a microscope 3D Explorer FLUR, and it helps us to understand the morphology, biology, behavior, and physiology of carcinoma cells with different responses to different therapeutic approaches. And finally, I would like to thank the Nanolife team for their readiness to help us always when we are contacting them. They help us in all applications, in all explanations, and I hope that we could just continue our collaboration. And finally, I'm very thankful to my team, especially Dragana and Alexander and Barbara, for providing me with these images and videos, and they're working with Nanolife microscope with great success, and they like to work with the microscope, and therefore I hope that we will continue to do it, and we have a lot of ideas how to improve and how to continue to work with the microscope. Thanks.
Thank you, Ira. That was very kind words and great images. Does anyone have any questions for Ira?
Actually, hi, I'm Johannes. The apoptotic cells, it really seems to distance the other cells, as if it would chemotactically remove the things to have space to undergo apoptosis. Or is this something common, or is this? I've never seen anything like that.
In literature, it is described when the cell would like to die, they would like to be separated from other cells. And therefore, it was, of course, we didn't expect that we will observe this apoptotic cell. And when we have observed that, then I tried to find literature about their behavior during apoptosis, but it seems that this is common for different cell lines, and especially interesting that this cell line is not very sensitive to the treatment, and therefore all other cells survived, and this cell wanted to die.
Right. But that must be an early factor during apoptosis, something that is very early on secreted by the by the cells that know that have started the program to undergo. But this is absolutely, yes, very, very, very strange, nice finding. Thanks.
Yeah. Thank you. And thanks to Dragana to her images and this finding. It was quite interesting for us.
Thank you. I think Branislav has a question.
Maybe if I can just add, I saw many apoptotic cells, and for me it was interesting that they remain you maybe see here that they remain in contact with one cell, even while they got separated. Somehow they do not lose a link, at least to the one cell nearby. Doesn't matter. It's really great how apoptosis look like with this system. And I forgot to thank to Nanolife team. Now thank you for reminding me that. But they know how happy I am with that. I wanted to ask you something just shortly. You showed nice mito stain, but you discussed just shortly lysosomes. Do you have any lysosomal stain tried so far?
Yes, we have tried. The quality of our images was not very good, but we will continue to do it. And
no, is it pH? Sorry, is it pH-like linked stain, or is it what was?
No, currently I cannot give you, because we have tried a number of different dyes and different stainings. And another PhD student, Bertram, did that, but I decided not to show that because the quality was not so good. And it was a great background, but generally we were satisfied. We have treated the cells with simvastatin, and the lysosomes were increased in their sizes, and we saw how they are released from the cells, and how the lysosomes moved to the proximities of the cells, and quite impressive videos and impressive images. Unfortunately, yeah, the quality was not good, but probably in future we could be able to show these videos.
Okay, thank you.
Thank you, Ira. Any more questions? Okay, if that's not the case, then we thank you for that presentation. We'll move on to Thomas then. You'll need to unmute yourself though.
Okay, hello. I try to share my screen. Hopefully it works. Let's see. Can you see it?
Yes.
Yes.
Okay, so it works. Good. Okay, so welcome. My name is Thomas Deicher. I'm from the Ludwig Boltzmann Institute of Osteology in Vienna. So in our institute, we are mostly interested in the microstructural composition and organization of bone, in bone development, bone properties, bone homeostasis. So actually most things related to bone. So we're an interdisciplinary research institute. So we have clinicians, physicists, and biologists, chemists working in this field. And so in my lab, we are the cell and molecular biology lab, and we are mainly interested in the differentiation process and the generation of osteoblasts. And yeah, Nanolife, unfortunately, we just had a demo version, so I can't show you like research we've done, but I think we acquired like in these 3 or 4 days quite a lot of nice images, and luckily we can collaborate with Dr. Riari from the Ludwig Boltzmann Institute of Trauma. They're getting this microscope, and yeah, hopefully we can make nice collaborations. But I'm just going to show you now the images and videos we required during this demo version, which was already quite impressive, I think. So, so we mostly use 2 model cell lines in our lab, which are mouse mesenchymal stroma cells, so-called T1 cells, and are mouse pre-osteoblast, MC3 T3 cells. Both cell lines can be differentiated into osteoblasts and even into osteocytes, if everything is perfect. So the experimental conditions is most likely that you let the cells grow or see them that they are about 80 to 85% confluent. Then you add ascorbic acid and beta-glycerophosphate to induce differentiation. And in 10 to 30 days, depending on the cell line, you end up with the cells which have produced an extracellular matrix and have deposited minerals. So sometimes the cells are really like they're coming up from the cell, cell culture plate and rolling up because they have such a strong matrix. So the device we used was this CXF. Yeah, and here this is just like D1 cells. They've just been induced for 1 day. And what you can see here is already a lot of things. So you can see plasma membrane ruffles, nucleoli, you can see the nuclear envelope, chromosomes. So here you have a dividing cells, and what we think might be calcium deposition. So that the main object, what I wanted to see with this microscope is if you can follow up the build-up of the extracellular matrix in these cells and see if calcium gets deposited. So we're not completely sure if these things are like calcium deposits, but they don't look like liposomes because they're too big and too irregular. So this was the first experiment. In the second, so these are D1 cells which have been induced for a longer duration, 5 days. So there are more cells already. They are still active, which was a surprise because we thought like normally they should stop proliferation and then start to differentiate, but it looks like they're still proliferating a lot. If there's more calcium deposition, I mean, this, as I said, it was just a demo. This we don't know. This we hopefully can investigate in the near future. Yeah, so this was overnight, a movie of 12 hours. I have to say the cells are really happy. Even if you watch them overnight, there are no problems. You can put the cells afterwards in the incubator and they're still growing and happy. So that is really, really good. So it's not really, the microscope is not really harmful to the cells. So next, yeah. So what we also tried, because I also want to see how the fluorescence looks like. So we transfect the cells with a lamin A GFP. So lamin is a component of the nuclear lamina and of the nuclear envelope. So what you can see here, you can see then the lamina and the nucleus stain. This is a transfected cell. To really for our purpose, I think it's a good tool to label cells. If you do like say knockout with shRNA and you have a GFP on your plasmid to see which cells has been knocked out, that's a good device. If you want to go really into details, structural details with fluorescence label proteins, I think the resolution is not great, but it's anyway not made for this. When what you can see here, what I assume these like bright spots might be transfection complexes. We did few chain transfection. Yeah, I think this is just from the transfection, the DNA or this DNA complex that sits on the cells. Yeah, this have been the MC3T3 cells. So they were just transfected the day before. Okay, so these are now images. So these cells have been just stills from videos. These cells have been induced for I think up to 10 days, 10 to 12 days. So these cells have not been induced, so there has no ascorbic acid and no beta-glycerophosphate been added. These cells have been induced with both, and there we didn't use beta-glycerophosphate because we wanted to prevent the mineral deposition so that you can see maybe better. And by looking now at the images for this for the presentation, I'm not sure, but it could be that because these are not induced, so they shouldn't produce like a a strong extracellular matrix and no mineralization. And in these two images, which have been induced, I don't know, but here, like these structures here, here, this might be already like maybe collagen fibers or from the extracellular matrix. So I don't know. As I said, it was just a demo, but this would be something very interesting to look into more details. And these structures here, so here it looks like more, they have like this more like a matrix, which is missing here, and here maybe it's a little bit blurry because maybe there's like mineral and this makes the images a little bit more blurry, or you can't see the details so well. Okay, and then I'm coming already to the last slide, which I just also showed my students because I'm teaching now a stem cell course, and they were very impressed also from the microscope. So these were just MC3D3 cells, again induced. I think I don't recall now exactly for how long. All of watched overnight, and if you see this spot here, you can see that like cells are eating up. I don't know what it is, maybe lipids sticking on the plate. So I think this was quite impressive. You look at this again here, and you can really nicely see all these membrane ruffles, and the cells are very active and still moving a lot. So here you can see that this is like really like endocytosis and eating it up. And then it goes also, you can also nicely watch mitosis with this microscope. And yeah, on the right you see a still, and yeah, this looks like this could be also some extracellular matrix. So that's our purpose, to watch like the generation of the extracellular matrix, and then of course compare like wild type knockout cells or treated cells with different agents, reagents, and yeah, maybe another side project would be see, as I'm also working on nuclear lamins, to see in laminae or in lamin knockout cells if you can see differences maybe in the lamina or in the nuclear envelope, and also if you overexpress mutants like a lamin mutant which is associated with porphyria, it should thicken the nuclear lamina. So if you can see this, and the third project which would be interesting is we have now a mutant which should affect also mitochondria. So to see if you can see like differences in mitochondrial dynamics. I think that's, if you work with mitochondria, this microscope is really good. So thank you for your attention, and questions are welcome.
Thank you, Thomas. That was nice to see these images again. It was right before Corona started, so
yeah,
it was the last demo I did on the field, actually. Yeah, but great. Any questions?
Not me again. I hope so. Yeah, I'm sorry. You mentioned a lot of lamina. Could you really? I presume you try to see lamina as much as possible. Is it possible to see lamina really?
What do you mean? With this microscope or with
this microscope? Only without any staining. Basically, it should be possible. It's like approximately 20 nanometer thick, just behind the nuclear envelope. So I presume thick lamina would make a nuclear envelope thicker.
Yeah, I mean, with the electron microscope, it's of course no problem to see it, and you can also nicely see the thickening. I mean, with the light microscope, it's tough. I mean, there's this these porphyria cells. They have a thickened lamina. There you can see it. So I don't know. I mean, I think probably the image from this from the Nanolife microscope comes from the nuclear membranes, I assume, or I don't know exactly.
You know, sometimes in fibroblasts, I see they are very good. Fibroblasts are stretched nicely. You can see everything there. Okay, I have the feeling that sometimes this nuclear envelope is thicker than I would expect, just if it is. I don't know. I just ask whether you have some experience.
With this microscope, I don't have any experience. Otherwise, it's hard to see. If it's really thickened, then I think you can see it also with a normal light microscope or like confocal. But otherwise, I think it's hard. As you said, it's like 20 nanometers or so, so it's not really thick.
Okay, thank you very much.
You're welcome.
Maybe just to jump onto that, our resolution is 200 nanometers. So if we were to see it, they would have to be bunched up together.
Yeah, that's true. Yeah. So from the nuclei, from the nuclear envelope is it that it's the membranes which makes the signal, right? Okay.
Yes.
So probably then I don't think you would see it because the membrane is not affected. It's just underneath the membrane, the lamina. So, but I mean, would be interesting just to check.
Yeah, for sure. Great. If there are no more questions, then we'll move on to our final speaker, which is Suzanne. Thank you, Thomas.
You're welcome.
Hello.
Hi.
Can you see my screen?
Yes, but we see the Teams, not the presentation.
What about now? Because now I see the presentation.
Maybe it's coming, give it a second. It's all white.
Yeah, I see a white screen.
Okay. 1 second.
Ah, yes.
Does it work?
Yeah.
Yes, great.
Oh, okay, sounds great. So my name is Susanne Fritsche, and as a PhD student, I'm working at the Technical University of Vienna, where we use Nanolife technology in our fungal lab. So our working group focuses mainly on the filamentous fungus Aspergillus niger and the metabolic engineering. So working with this type of organism, the fungus, we, especially when it comes to this microscoping technique, we have the challenge of a fungal cell wall, so which consists of mainly glucans, chitins, and glycoproteins. But nevertheless, we do see differences in the refractive index on parts of interest. So we are able to use this live cell imaging tool to explore the germination as well as the nucleus of our organism. So I want to give you a short overview about how our setup with the fungus works. So we have those spores, and they're suspended in our media, and we supply it with glucose to initiate growth. And from a dormant spore to the growth of a hyphae, it takes about 6 to 10 hours. So we use our incubation chamber with airflow and optimal temperature set. So what you can now see on the video is the transition from a dormant conidia to hypergrowth. So the video is composed of 230 frames here, which is generated during 8 hours. And after the spores break dormancy, they swell to this size by water uptake and go into isotropic growth. And this is then followed by polarized growth, where the germ tube then forms. So we are interested in the morphogenetic events on the site of newly synthesized cell wall. And we are able to see changes in the refractive index at locations of interest, which are here. And with the tool, especially RI rendering and the digital staining, we are able to see a formation of a ring structure, which is in the picture here. So the isotropic growth with the ring formation, which is only seen with the tools, the software provided. And this kind of ring structure directs the tubing to one side of the spore. So to get a deeper understanding of this process, we will focus on proteins involved in tube formation by fluorescent tagging and then tracking while live cell imaging. So that is an outlook on that topic. On a genetic level, it is also in our interest to investigate how nuclei in Aspergillus niger influence genetic heterogeneity. And in the first place, we want to observe if our fungus is predominantly mono or multinucleate. And to do so, we are now generating nuclear localized fluorescence reporters. And with Nanolife, we want to observe the nuclear conditions during the growth process. So with this outlook, I want to already conclude my presentation, and yeah, hope you enjoyed the insight in our research strategy using Nanolife. Thank you.
Thank you, Susanna. Yeah, it's nice to see a different application as well. For us, it was quite surprising when we did this demo because we didn't expect it to work this well. Yet here we are. So.
At university in our building, we have a lot of research going on in with fungi, and people are interested to also now see them growing like on screen and live, which is very interesting for them to see also, and which works also really, really well. So now, yeah, we are very interested, like after having those reporters and the fluorescent text, how the pictures will look like and how we can track and get to our results.
Thank you. And yes, for those who don't know, so it is a label-free microscope, but we also have 3 fluorescent channels that can be overlaid on top of the images to add more information to this label-free information. Great. Are there any questions for Susanna?
Actually, how large are the conidia here?
2 micrometers.
Okay, right. Thanks.
Starts, and then they swell up to double of the size. Yeah.
I hate to ask again, but I tried once germination of Bacillus cereus with colleagues here from hygiene, and we had troubles with this because they are swimming, of course, the spores. Then I used just agar. We put them in agar and then add media over that. It was great. I can send you this image. It looks perfect. You can get even in the 3rd dimension nicely development of bacteria just by using agar.
Okay, that sounds great. Would like to see those pictures and the setup.
And I would like to have protocol as well. To be able to suggest to other customers. Yeah.
Okay, I sent this video to Sanela or Kulsum, and it looks similar to your germination. Just like after 24 hours, I get the whole field full with bacteria, and with the new macro, you really get them in the same. It's really good. I was quite surprised that works with agar very well. And maybe a small tip, I don't know if anyone here works with, for EBD, I use the same dishes since September. I recycle them all the time. So it works very well with one package. I just add some NaOH and then I wash them with acid and wash them very well with water, and then I add alcohol to be sure that this is clean, you know, no germs, and it works well.
Okay,
just
good
a small
to know.
tip. Yeah, you can save some money.
Excellent. We don't have a partnership with EBD, so we're fine if you reuse them.
We won't tell them.
We won't tell them. Okay, wonderful. So thank you all for those presentations. I'm just going to end with a couple of slides just to show you, because all of these users are using the manual version of our microscope, so single field of view. And since then, we have some developments that have been made at Nanolife. So I just want to show you just a few slides to give you an idea. So the first question was that we had from many people was that the field of view was too small. And due to the way the technology works, it's complicated to change the objective. So what we did instead is we have a grid scan possibility. So now you can combine, as you see, this is a single field of view, normally 90 by 90 microns. And we can then do grid scans and to create larger squares to get much more information, up to 1 millimeter squared. So it's a lot of information. And here you notice that there's some pauses and you see the cells get colored. that's a slight teaser. You'll just see it happen here. It's a slight teaser for our latest analysis software that we have associated to our microscope, where you can actually segment these cells and get information on the single cells and the population as well. So just to keep in mind that even though we're working with, because you're working with grid scan, even though we have larger fields of view, the individual resolution is always maintained since it's the same imaging technique. So it's a very smooth, smooth way of getting more information. So here it's again, it's highlighting the segmentation again. And as you can see, it works well with sparsely spread out cells. But as these cells are increasing in confluence, as they divide and proliferate over time, so here is a 90-hour time lapse, you will see that it's still a very precise segmentation, even though the cells are all dividing and spreading around. And that's how we actually got these metrics of confluence, because the cells were segmented, and compared to the area, we could then get the percentage of coverage of these cells. So that's just one of the metrics that we can get. We actually have many different metrics, such as the cell area, the cell compactness, the extent, so the granularity as well, the perimeter. These are all information that it's all morphological information that can give you information on the general health of the population of the cells. You can also get the information for the single cells. So you can then get averages in a population and see, for example, what the tendencies are of the cells in that population. One parameter I'd like to highlight the most, it's the dry mass. Sorry, my computer is lagging a bit, so it's a bit jumpy. I apologize. But with the dry mass, this is actually a very interesting metric that we can get, because with our microscope, we're picking up on information of the refractive index. That's the output that we get, is a quantitative value. So directly from the refractive index, you can calculate the dry mass of the cells. So you can see how the density of the cells is changing over time and how the internal composition is changing. And that gives an interesting information as well for certain studies. So yes, that's just a short preview of what can be done with Nanolife imaging. You can see it can be used in multiple fields of view, as we saw from the previous presentations. And we are always excited to see what else can be done with this system. So that's the end of my part of the presentation. I'm going to leave the floor open to any questions if people have. If they have questions, if you have questions on your specific applications as well, please let us know. And I know a few of you have requested a virtual meeting with me after this presentation, so I'll be contacting you in the afternoon to arrange this, so we can then discuss further as well if you have more information on the technology, if you need more information on the technology or on your own applications. So that's it from me.
Branislav raised the hand, but no, it was from before.
No.
It was from before.
So, are there any questions?
I think everyone is speechless.
Must be. I mean, the four of you summarized it quite nicely. I didn't even need to speak at the end. Okay, if that's not the case, you're always welcome to contact me by email at any point if some questions come up. And I'd like to specifically thank the four presenters as well for their time and for these beautiful presentations. It's always nice for us as well to see what our customers are doing with the system. It's very exciting. So thank you all.
Thank you very much.
Thank you.
So I've recorded this. I can send you a link to the recording if you'd like to download it or share it with some other people. Feel free, since we have permission from everyone.
Okay, thank you very much.
Welcome. Thank you. Okay then. Thanks, Sarah. Thank you, everyone.
Thank you. Bye-bye.
Have a nice day. Thank you for your collaboration.
Bye.
Maybe you can stop recording.
Yeah, I'm going to stop the recording. That's what I was looking for.